I'll give everyone a moment to settle down, but I'm very excited to introduce this afternoon's session around advancing CMT research projects into industry. My name is William Motley. I'm a physician scientist and neurologist by training, and then after training joined the biotech community in Boston. I've been at a few different companies, and I'm currently at a company called Rapport Therapeutics, where I lead the lead program. It's a wonderful session this afternoon and something that I'm really excited to share with the group, and to moderate the session. It's an exciting session that I think gets at the heart of what we're trying to do as a community and at the foundation, and that is translating scientific discoveries into approved therapies. Three talks really form an interesting cross-section, I think, of and review of therapeutics development, and are stories that should really inspire us and motivate us in the CMT field. The first is a story of discovering and developing an approved gene therapy. And there's so many lessons to learn from successes in other diseases. So this is a therapy for SMA. And SMA, I think there are a lot of really interesting and important lessons from this. There are multiple approved therapies. There's always going to be a need for multiple approved therapies in each disease, and with complementary approaches. Foundations played a critical role in the development of a few of these therapies. And the success begets success, and now there's a really rich and robust pipeline in SMA. The second story is a discovery made in fly in 2012, fly and mouse, and then really a story of a small venture firm in Boston taking an interest in this exciting science, building a company focused on developing small molecules for this target with broad applications. It wasn't an easy target to drug. I think it involved a lot of persistence and experience, drug developers going after it. I think early success, publication of structures, and now there are tons of companies working to target SARM1, and it's an area of incredibly exciting research. And finally, a story about early drug discovery effort, but a story that started with the identification of GARS mutations as a cause of CMT in the mid 2000s. Really focused work from a series of labs on understanding the science underlying that pathology, and a few interesting and exciting therapeutic approaches that I think have traction, one of which we'll talk about today. So the three speakers who I'm excited to introduce are Brian Kaspar, Aaron D'Antonio, and Maria Landinez. Brian will speak first. He's currently the Chief Scientific Officer at Insmed. Brian joined Insmed in August 2021 when Insmed acquired a company that he co-founded, focused on life-threatening and debilitating disorders of the central nervous system, musculoskeletal, and ocular systems. Dr. Kaspar brings more than 20 years of experience in gene therapy and development and manufacturing, including leading the development and commercialization of gene therapy for SMA. He trained at the University of California, San Diego, and the Salk Institute for Biological Studies in La Jolla, California. He later served as professor and endowed chair of studies of pediatrics at The Ohio State University and Nationwide Children's. And he has co-authored more than 100 scientific publications in high-impact journals with specific expertise in neuroscience and AAV gene therapy. He's the co-inventor in numerous patent applications, which led to the creation of biotech companies, including Avexis. Brian, thanks.
Thank you so much, William, for the kind introduction, and it's a real honor to be here today. Thank you for the invitation, and as you hear my talk, you will say, Brian, talk less, because there's some videos here from our preclinical work to seeing the potential of transformative therapies. And I had the privilege of leading one of those efforts throughout my career, from academics into industry, and then to see this from the bench to the bedside to commercialization. I just want to say there is no one more humbled about the power of medicine. And I think that when we get things right, and it's very challenging to develop a drug. And let me just say, as CSO of Insmed, we're not working in CMT. We are interested in CMT, and when we want to partner, there's opportunities to reach out to me. So I just want to open that up. Of when you have something, please reach out to us. I will do it pro bono. We will bring it into Insmed if we can. So there's just opportunities here because when you see this story, you'll see the true magic of the power of medicine. So I'm going to be talking about a FedEx truck called adeno-associated viruses. This is a gene delivery. It's been out there since the 1970s. It's been in multiple hundreds of clinical trials. It's ultimately safe. This 20 nanometer viral particle with DNA stuffed on the inside is the FedEx drug. It's extremely stable as a drug product, and when it enters a non-dividing cell, it doesn't integrate into the host cell chromosome, but it forms an episomal concatemer, which is extremely stable. So if the cell is not dividing, this thing can express for years, if not indefinitely. I can't say indefinitely because we're still going, but we have now clinical evidence that you'll hear of well over 10 years of experience of stable gene expression. So essentially, what this virus does is it attaches to the outside of a cell, it gets on the inside, it goes into the nucleus, and then it dumps its DNA payload. So you control what we put in. So your favorite gene of interest, IgMHB, with a promoter, could fit into an AAV, as you heard yesterday. When that cell actually gets the gene, it becomes a factory and starts producing the protein or the gene of interest. So we can do gene replacement, we can do gene knockdown, and even gene editing with novel insights within the field. So my field really started in the late 1990s at the Salk Institute, and my mentor, Fred Gage, Rusty Gage, was a real pioneer in CNS translation. And one of my goals was to try to see how could we deliver to the brain and spinal cord efficiently. And throughout the years, or over the course of a decade, we actually made a seminal discovery, and that is an AAV9. Instead of drilling holes into the brain and dropping needles into the spinal cord, we actually discovered an AAV serotype that could cross the blood-brain barrier after being placed into the vascular system. So an IV exposure of an AAV9 crossed the blood-brain barrier and targeted cells throughout the brain and spinal cord at really unprecedented levels. We targeted neurons, we targeted motor neurons, and you're seeing here on the cover of Nature Biotechnology, motor neurons from the cervical all the way down into the thoracic region of the spinal cord, along with other cell types, such as astrocytes. So it was a real game changer in the field, something that we had been dreaming about. And this led us to think about a one-time administration to target these non-dividing cells where we targeted surprisingly over 90% of the motor neurons throughout the spinal cord. And this kind of opened up the idea of thinking about a disease called spinal muscular atrophy, which is an orphan disease, devastating, resulting in motor neuron loss and progressive weakness. And it's the most common cause of genetic infant death. So incidence of 1 in 10,000 live births, and it's caused by an exact gene that we know, the SMN gene, and we'll talk a little bit about that. But this is Wernicke-Hoffmann disease for the clinicians in the audience. It's called also floppy baby syndrome. Not good. These babies can't lift their heads, can't roll over, will never sit up unassisted, will never roll over, will never talk. And as you'll see in the natural history, death almost certainly by 20 months of age. Here's the natural history. By 10.5 months of age, 50% of these kids are either on a tracheostomy or have passed away. By 13.6 months of age, 25% have passed away. By 20 months of age, only 8% of these kids are event-free. So it's devastating. I grew up in my training in an area where doctors told families, take your baby home, love them, they will die, make them comfortable. It just seemed wrong. And fortunately, there's a number of drug companies, including Biogen, including now gene therapy, including Roche on a small molecule, that have made a real change in this disease. So essentially, normal individuals, here's the genetics. Essentially, in the bottom left panel is the strategy that Biogen and Ionis were taking to increase a backup gene called SMN2, to increase the numbers of SMN2 to create a functional protein. These patients have lost SMN1. Our approach was gene replacement. Let's just replace a gene, create a lot of it, and see if that worked. So here we are, we're utilizing the capsid shell AAV9. What we do is we gut the inside of the virus, we put in our ubiquitous promoter. It's this continuous promoter that drives strong expression of the SMN gene. So we're truly producing exactly what these patients need. It has a polyadenylation signal and all of the aspects to essentially make the motor neuron, our target here, become the factory for what it's missing. So we went into the mouse model of the disease. It was developed at the Ohio State University by Dr. Arthur Burgess. This mouse model is called the SMA Delta 7 mouse. It dies at 15 days of age. So we know pretty quickly whether our drug is doing its job. And a one-time IV exposure into these mice actually led to these animals, median survival passing 200 days. It had not been seen in this field at this time. So really striking results. But what works in a mouse, does it work in a monkey? We went into monkeys, we targeted motor neurons, we felt pretty excited by an IV exposure. These mice, not only were they living longer, but they were running around the cage looking like a normal mouse. We worked, externally with Dr. Mark Rich at the Wright State University, a clinician scientist, and Arthur Burgess to look at the neuromuscular junction. And we actually put electrodes into these mice and showed that they were firing the right action potentials at the motor neuron junction. They were making the right neuromuscular junctions within the muscle. So this looked like we had corrected these animals. But this was a mouse. And cancer has been cured a lot of times in a mouse. And we kind of put the same pressure on us. A mouse can be cured, but can a human be cured? So we actually, with Dr. Burgess's lab really leading the charge, us being a real strong part of this, we developed a pig model of SMA. And let me play the first video to my colleagues who are playing the videos. If I can play the blue video here. And I think the big discovery here is that you can train a pig to run up a ramp to eat a marshmallow. So that's our big discovery here. So the pigs will run up a ramp here. In the second video, we actually developed an shRNA in which we knocked down SMN. And if we can play that video, it's a hard video to watch, folks. This is SMA in a pig. We created SMA in a pig. That pig can't get up the ramp. We took good care, and trust me, that pig got even more marshmallows. Let me show the next video, the one on the bottom left, gentlemen. If we can play that video. This is when the pig developed weakness, we actually treated with AVXS-101, our gene therapy, to replace gene therapy. Now, we didn't erase the problems completely, but gosh, that pig got up the ramp and actually kind of improved. Then we played the scenario of what if we had newborn screening and we could treat that pig before the signs of onset and symptoms. And if you could play that last video. That pig essentially doesn't have SMA, even though we created SMA on that pig. And it ran up the ramp. There might be subtle signs, but that pig almost doesn't have SMA. Really powerful and really led to our confidence in starting clinical trials in a safe and what we believed would be an effective manner into patients. So 2 cohorts, we started working with the FDA, minimally effective dose to a therapeutically effective dose. And this was really led by my friend, my colleague, Dr. Jerry Mendel, Dr. Zarif Sahang, Dr. Samia Al-Zaidi, part of Nationwide Children's. And, I'm going to jump through to basically say, I told you the natural history. We had 15 patients in the clinical trial, low dose, higher dose. 15 out of 15 patients surpassed that 20 months of age, where only 8% of patients would have been expected to have survived or be event-free. Now, patient 2 had a situation, they needed to go on a vent, but that was at month 30. Really remarkable results. When we look at Dr. Rich Finkel he used to be at the University of Pennsylvania, developed a CHOP intent, so an infant test of motor function. The top score is 64. We had 2 patients, some of the earliest dose that achieved a 64 point. So these kids didn't have SMA. They developed normally. One of the patients, as you can see along the bottom, one of the oldest patients didn't necessarily respond at the same response rate, but was stabilized. You see that dotted line at 40. Let me just say that SMA patients with SMA type 1 never achieve a score above 40. Here we have 11 of 12 patients achieving scores above 40. Really remarkable results. Numbers, and we worked with the FDA, and I know I'm on timing here, so, but I want to show, in what I said is I shouldn't talk at all. I should just show these videos. A reminder, natural history, these kids don't roll. They don't sit. They don't talk. They don't eat. They don't talk. And I think one of the things that we did back in the day at Nationwide Children's, at Avexis, the company that I started, which was acquired by Novartis, was to capture this information so that we worked with the regulators in the aspect that you can see our patients. Patient number 9 here is showing fine motor skills, sitting unassisted, just things that are not in the textbooks for spinal muscular atrophy. So I should have been faster because I'm running out of time. But Will, if you give me just one or two more minutes, I'll get through here because I think this is just a remarkable story and humbling on the power of medicine, on the power of seeing things that shouldn't be happening. This is Dr. Linda Lowe's, our the physical therapist at Nationwide Children's, and you'll see one of our patients, patient number 10, who was one of the earliest diagnosed. And she's really lucky because she gets a nice little hug from Mateo here. And then he's running back to his dad there. And, Mateo is looking and continues to look like a normal boy going to school. He's 9 to 10 years out, folks. And I hear from the family, he's thriving. A one-time gene replacement, a blood infusion into his arm, and you'll see what he does here. He does what any little kid wants to do, push elevator buttons that frustrates us when we're checking into our hotel rooms. One of the things I think in gene therapy is manufacturing. We invested a tremendous amount. We continue to invest in manufacturing. Gene therapy, one of the struggles is getting this right. It's how do we make this? How do we make enough of this? How do we keep the costs down? We can't continue to have $1 million drugs. Don't shoot me. I'm not the messenger of drug pricing here, but these are expensive medicines. It's difficult. We're making progress as a field. This was a start in controlling, understanding, and having the quality control of the product. That product, batch 1 is the same as batch 3, as the same as batch 333. The field is struggling right now. But I think some of our experiences in getting ahead of this has been a real savior towards SMA of making this a reality commercially towards patients. So AVXS 101, we actually scaled this from a phase 1 clinical trial and got this towards a commercial scale setting. Took that commercial scale setting, and the FDA really asked us and worked hand in hand with us to basically say, can you make this? And actually, here's the commercial product overlaid on that phase 1/2 clinical trial. It essentially parallels our data that we had. Very gratifying here. So we applied in November of 2018 for a biological license application. It was approved in 2019 by the Food and Drug Administration. It's now approved in over 50 countries. Novartis is leading the way. Zolgensma is a drug that has now treated over 5,000 patients with pretty impeccable safety profile. I'm gonna just, if we can have the sound on this one. I know it's after lunch, people might be tired, but this is, I feel good. Really amazing here. It's been sorry, truly humbling for me to lead projects from bench to bedside, to lead the fantastic teams from clinicians to manufacturing personnel to all of the things to make a drug move forward. And to see the result here, it's just truly a goosebump and humbling to me. We hope to continue to do the same. We hope that in the CMT, if there's a gene therapy, please reach out to me. I'm happy to help. We're happy to partner because this is a field that I've paid attention to, lots of people to pay attention to and think forward to, but I'll stop here. Well, thank you so much.
Be an opportunity at the end for everyone to ask questions, but maybe one question now for Dr. Kaspar.
All right.
Gary?
Terry.
I was just wondering how the manufacturing portion worked out. Like, how did you go from clinical to PPQ batches and maintain that? And how did you work with the FDA on that portion?
Yeah, so we had to change because that was back in the day. I'll talk a little bit. I'll be quick because of time, but back in the day we were in hypersex. So
Okay.
that satisfied a phase 1 clinical trial that couldn't satisfy rest of world. So we had to transition. We kept an adherent cell process because we had been adherent. We moved to what was called the iCELLis unit. Basically, it's a football field stacked in a big bioreactor. We worked with the FDA. We showed comparability. One of the things, the drug probably would have been approved 2 years earlier, except for we lost stability of the drug because there was a quality control issue. And therefore, FDA made us go and do the phase 3 trial. But they also worked with us to truncate that phase 3 trial. So it was a hand in hand. A lot of people argue with the FDA that they're bad. They're actually doing their job, and they did the right job in this case. And we worked with them hand in hand to show comparability, and then we went into patients, and you saw those numbers were identical to the phase 1 trial. So many drugs, people make changes in the manufacturing process. So one of the things that we've learned now in the day, and the field has grown in gene therapy, that now how we think at least at Insmed is we're almost at scale when we start the clinical trials, but that was back then. And I think actually a lot of things start academically and then they transition, but that's where you can get into trouble.
And that's exactly what I was asking, because in the end, when you go from research grade toxicology to CMC on your phase 1, 2, it's just like, just get it in there. But that's the fault, because then once you start going from a clinical to the PPQ batch is like climbing Everest.
And so now we're trying to get ahead of it. Now we've got really more sophisticated CDMOs. The field is actually matured a lot. We're not there yet, folks. Let me just tell you, there's still a lot of work to do in the gene therapy field. But I think there are very good stewards who are thinking about this in the right manner here, so that we don't necessarily have these 2-year delays of getting a drug out to patients.
Thank you, and congratulations.
Thank you. Thank you.